Hello, I am Agaz Wani. I am a postdoc in Dr. Din's lab at University of South Florida. Today I'll be talking about quality control steps that we need to do in epigenome-wide association studies. So I'll be talking about check for failed assays, mislabeled and contaminated samples. Then I'll be talking about dye bias correction and normalization, followed by that removal of low quality probes and samples. And finally, I'll be talking about adjustment for batch effects. So we need to install and load some packages. We will be using EWAS tools to perform quality control check. So the first thing we need to do is to load the IDAT files, that is the raw data. Each sample has two IDAT files, that is green and red channel. We also have sample sheet that comes with the raw data that looks something like this, where we have centric site and centric position. We have the sample name. So we need to use this sample sheet to read the IDAT files. So we are reading the IDAT files. It will take a few seconds. We are almost done reading the IDAT files. Then we will check if we have all the samples on the sample sheet and the methylation data. So the next step we need to do is to evaluate 17 quality metrics that is recommended by Illumina. We will remove the samples that fail this check. So it looks something like this, where we have the sample ID, that is the combination of Centrix ID and Centrix position. It will be unique for each sample. And we will have the information here whether the sample failed or passed this step of quality control. So if the sample failed, the value will be true here. Now we need to do the genotype check that will help us to find the mislabeled or contaminated samples. So it will give an outlier score. If the outlier score is greater than -4, it is recommended to remove that sample. So we have the information here. We have written it to the file where we have the outlier information. If it's greater than -4, it's recommended to remove the sample or flag the sample so that you can track it in the downstream analysis. And we have the donor ID and the number of samples with the donor ID. So in case of longitudinal study, where we have more than 1 time points of data for a participant ID, it will have the same ID for that participant. So as we have unique samples, so all these IDs are unique. In cross-sectional analysis, we need to remove the duplicate samples or IDs. Then finally, we will write this data so that we can use it in the next quality control steps and downstream analysis. In this part, we will be performing normalization, dye bias correction, sex check, and combat adjustment. So we will install and load the packages. We will use some man packages like MinFi, SVA, and CPG association, R packages, and some helper R packages to make the process easy. So first we will load the IDAT files and the sample sheet that we saved in the previous file. We will need some more information like age and sex and PTSD that we will use in the combat adjustment. So we will load the data first. As you can see, it is loading the red and green channel data, and it will perform dye bias correction because we have data from 2 different channels, that is red and green, and it will perform SS-NOVE normalization. There are various approaches, but in this part, we will use SS-NOVE. Followed by that, we will do sex check. We are almost done. Now, in the next step, we will perform sex check. So the data in the phenotype file and methylation data should match. So if there's any sex mismatch between those two files, we need to remove the sample. So it means to say that there's some issue with that sample. So we don't want that sample in the analysis. So we also need to have p-value, beta value, signal A, that is unmethylated information, and signal B, that is methylated information, to perform some other steps. So we will also save this information so that we can use it in the future steps. We will also save this updated phenotype information to use it in the next steps. So once we have saved this data, then we will clean the environment and load the data again to perform some other quality control steps. So we are almost done with this step. Now, when we have saved the data, we will load the data here and do the check, that is to remove the low quality probes and samples. So if there's any sample that has greater than 10% of the missing value, or any probe with greater than 10% of the missing values, we will remove that. As you can see, 0 samples has been removed, but 1585 CPGs has been removed due to higher missing values. Next, we need to remove the cross-hybridized probes, those probes that match more than one location in the genome. So those are recommended to be removed. You can refer this paper where it has known probes that are known to be cross-hybridized. We will remove those probes and we will save the data again for the future use. The next step, very important step we have to do, is combat adjustment. So it will remove the batch effects that are in the data. But one main thing we have to keep in mind, that we have to save the variation or preserve the variation for the phenotype of interest. For example, in this case is PTSD. We also have to save it for sex and age. So we will build the matrix for that and use that matrix to preserve the variation in combat adjustment. So for combat adjustment, we have loaded the saved data and we have loaded the saved phenotype data, and we have removed the samples that failed QC. Now we will order the samples or sort the samples so that they should be in the same order in the phenotype file and the methylation data file. So we will have sex, age, PTSD, chip ID, and chip position. We will be using combat adjustment to adjust for the batch effects for chip ID and chip position, and save the variation for sex, age, and PTSD. We also have to remove the missing values if there are any missing values in the phenotype information. So we will remove those samples. The other way is to include the samples, the information in those samples. But here in this part, we will remove those samples. Now convert the columns such as chips and chip position as factors, and PTSD and sex as factors, so that they can be considered as categorical. variable. So here is the matrix we have designed where we have PTSD agents X. So the variation for all these variables will be saved when the combat adjustment will be performed. We are performing log transformation so that we have a uniform distribution of the data, and we will impute the missing values in the methylation data because combat will not work for the data that has missing values. And it's imputing right now the data. Here, the first adjustment will be done based on chip ID, and the data, the outcome data will be used for the next adjustment that is based on the chip position. So you can see as we have 3 chips, so we have 3 batches that are being adjusted for the batch effects. The data is being adjusted for chip position as we have 8 positions on the chip, so we have 8 batches. After combat adjustment, we will be again converting the data to beta transformation, where it will have the data in 0 and 1 range. Converting the data back. And inserting the NA values back because we imputed the data for combat adjustment. So we need to put the missing values back. And we will finally save the data that is combat adjustment normalized. After sex check, we will save the final data so that we can use it in the downstream analysis. Thank you.
Hello. So for the second part of our pipeline journey, I will talk about how to run the analysis, the epigenome-wide analysis, and what we need for us to run the epigenome-wide association studies, and how we can get what we need from using our pipeline. So this is the general structure of the pipeline. Aras kindly talked about the initial quality control steps, which are described in script 1 and script 2. And I will talk about script 3, 3.1, 3.2, 4, and 5. Briefly, the EWAS is done by using script 4, but in order to run the EWAS, we first need some preliminary scripts to estimate cell proportions, principal components, and smoking scores. Cell proportion estimation will be done using script 2. This script needs normalized and QC beta values and phenotypes that was done by script 2 to calculate cell proportions CD8, CD4, neutral killer cells, B cells, monocyte, and neutrophils using EpiDish R package and IDOL reference panel. And for the output, we will get cell proportions. The script 3.1 is actually not a mandatory script. So we're going to use it if PCs from GWAS are not available. So here in script 3.1, we still use a normalized and QC beta values from script 2 to calculate methylation principal components using CPG sites that harbor SNPs. And this procedure was described in the paper of Barfield et al. And as an output, we will get methylation principal components. Then we're going to move to script 3.2, which uses 39 probes to estimate a smoking score. And so this EWAS, our main analysis, uses normalized QC and combat adjusted beta values and phenotypes from script 2, as well as cell proportions from script 3, principal components from script 3.1, and also smoking scores from script 3.2 for running smoking sensitivity analysis. And here, after running the script 4, we will get our output summary statistics that each group can share for us to run the meta-analysis. And finally, we have the script 5, which we use to calculate cell-specific differential methylation. And this script identified as cell-specific differential methylation using PTSD as the main effect and agent sex as covariates. So at the end, we will get summary statistics for each calculated cell type. So how we calculate cell proportion estimation, principal components, and smoking scores. So for cell proportions, we use Epidish R package using Epidish function with robust partial correlations mode. Here we use an EPIC-specific reference panel described in the paper Salas et al. This reference panel is kind of advanced than the previous ones because it includes more samples, 37 samples from a diverse background. It includes 6 African American, 2 Asian, 21 European, and 8 mixed ancestry. And it also has females and male smokers and non-smokers and has a wide age range. And then we calculate methylation principal components. If principal components from GWAS are not available, we use CPGs within 0 base pair of SNP, and we use methylation PC2 and 3 that are shown to correlate most with ancestry. The principal component 1 has been shown to correlate with cell type, so we don't include that because we already include cell proportions that we calculated from Epidish package. And finally, we calculate smoking scores using 39 smoking associated CPGs. That's way much better than using self-reported smoking scores because it helps us to distinguish, let's say, Jack, who smoked 1 cigarette per day, and Amy, who smoked a pack of cigarettes per day. And finally, we run our EWAS using the normalized and combat adjusted data, and we test the association between PTSD and CPG sites, adjusting for cell types. Here, the important thing is we only include 5 cell types and leave out neutrophils, which has the highest proportion, because at the end, the sum of all 6 equals to 1. So it doesn't make sense to include all 6. So we exclude neutrophils. We include principal components either from GWAS or methylation data, age, and sex if available. And then we run a smoking sensitivity analysis, including smoking scores as a covariate. Then we can run sex-stratified analysis if applicable. Those include sex-stratified and ancestry-stratified analysis. And finally, we will have summary statistic that will be used in the meta-analysis. And finally, for cell-specific differential methylation, we use R package toast to identify cell-specific differentially methylated CPGs that associate with PTSD. The idea with that is each individual may have different cell type proportions, which may mask the associations. So in using this method, we will have cell type, we will have CPGs that also associate with PTSD in each 6 cell types. So to sum up, using this pipeline, we will get EVA summary statistics for our main model, smoking adjusted model, and sex stratified model, and race stratified model if applicable, and also summary statistics for each cell types. So I would like to once again mention our PIs, Alicia, Caroline, Monica, and Mark, and our analysts, Agaz, Andrew, and Adam, who help making this pipeline. Also, if you want to join our calls, our regular calls are on the third Thursday of the month. Oh, thank you.